Welcome! Today we are discussing how the genetic makeup of a glioblastoma or GBM determine its relationship with the immune system based on research by Ryan Miller and Anita Hijalman. We will explore why breaking the IL-1 beta feed-forward loop is a break-off for some genotypes but ineffective for others. We will see how two distinct pathways, the proneural or PDGFB and the mesenchymal NF1, interact differently with the inflammatory microenvironment. TUMs, or tumor-associated macrophage, are not just bystanders. They can make up of 50% of the total tumor mass. These cells originate from either resident microglia or BMDM or bone marrow derived myeloid cells that are recruited into the brain. Instead of fighting the cancer, these macrophages are hijacked to produce pro-inflammatory factors that drive tumors' growth and create an immunosuppressive environment that shields the tumor from the immune system. To study these interactions, researchers use RCAS NTVA system, a lock and key viral method that allows for precise genetic engineers of neural stem cells. By elevating the PDGFB oncogene, we model the proneural subtype by silencing NF1 alongside TRP53 and 10-PTEN, we model the mesenchymal subtype. This allows us to isolate how specific mutation change the tumor's immunological changes. Language. The results are striking. Genotype determines survival outcomes. In the PDGFB-driven model, the tumor has a high resilience on IL-1 beta. Knocking out this cytokine extends survival significantly. However, in NF1 silenced model, there is no change in survival after IL-1 beta knockout. This is because the NF1 model has constitutively active or always on level of NF-κB, making it independent of external triggers. A critical finding is that the fuel for the tumor, IL-1 beta, does not come from the cancer cells themselves. Single cells RNA sequencing and IBA1 staining confirm that BMDMs and microglia in the microenvironment are primary source. Neoplastic tumor cells have minimal expressions of this cytokine, meaning the tumor relies on a paracrine signal from its neighbors to drive. The proneural circuits, or a vicious cycle of inflammatory sites. The IL-1 beta binds to IL-1 R1 on the tumor cells for phosphorylating NF-κB or NF-κB in the GBM tumor cells. Tumor cells with active NF kappa B secretes MCPs or CCL2, CCL8, CCL7. This increased MCPs drive a massive BM-DM infiltration into the tumor. Thus, BM-DM infiltrated secrete IL-1 beta, which IL-1 beta binds to IL-1R1 in a vicious cycle which wouldn't stop. The mesenchymal bypass high basal activity ignores external triggers. The GBM tumor cells overload constitutive activation of NF-kappa B. It's increased TAM and BM-DM. Independent output basal NF-kappa B drives MCP levels strict via internal signaling, driving BM-DM infiltration. In this paradox note, human mesenchymal GBMs have higher IL-1 beta levels, but the tumor does not functionally depend on it for growth. Severing the loop extended survival PDGFB-driven models, or TAMs, secrete IL-1 beta pathway and anti-IL-1 beta antibody and IL-1R1 antagonists to block the arrived IL-1 beta pathway to the GBM tumor. It causes TAM reduction. It's significant decrease in IBA1 plus macrophage percentage. Immune activation increases levels of granzyme B, enhances cytotoxicity, and the decay point relief reduces levels of immunosuppressive checkpoint PD-1. In the IL-1 alpha compilation, a delicate therapeutic balance. The condition 1 WT mice baseline not change survival benefit, and the condition 2 IL-1 beta knockout only extended survival. Deleting IL-1 alpha alongside IL-1 beta reduced microglial proliferation and potentially enriched therapy-resistant glioblastoma stem cells. Precision target of only IL-1 is functionally required. We have some variables. The male and the females are different response to this hormone therapy, and the female have increased of survival than males, and the other different our models is a young mice, while human GBM typically present at the age 65 plus 65. And in the human, we have intratumoral heterogeneity, or single patient hosts multiple transcriptional subtypes simultaneously in standard therapy like dexamethasone proactively alter IL-1 beta levels. At the end, the TMA tumor axis genetic dictate immunological language effective clinical immunotherapy in GBM will fail if applied universally. The microenvironment cannot be treated independently of the tumor score mutation. The precision immunotherapy requires genetic sequencing. We must identify the tumor circuit architecture before we attempt to break up it. Thank you very much. See you next time.